The part of the microscope that's used for the viewer to look through, and it contains a lens of 10 times the magnification, is called the eyepiece. That would be part 1. Part 2 of the microscope are the various lenses that you actually turn, and they're used to magnify an object either 4 times, 10 times, or 40 times, and that is called the objective lenses. The actual platform that the slide is placed on is called the stage, and that is part 3. The bottom that supports the actual microscope is part 4, and that is called the base. The way you actually adjust the microscope is you turn this knob and it raises and lowers the stage, and as it does, it focuses the object that's on the stage. It's called the coarse adjustment, and it's part 5. Once you use the coarse adjustment to get the object into view, then you use this knob to fine-tune it to your particular viewing site, and it raises and lowers by really small amounts by the turns, and it's called the fine adjustment. Part 7 of the microscope is the stage clips, and these are used to hold the glass slide in place. You just lift them up a little bit, slide that microscope plate underneath there, and then put the stage clips back on it, and it holds it in place. And the last part of the microscope is the thing that you hold it by along with the base, and that is the arm. It's the curved part of the microscope, and that's what you carry it by. You don't carry it by anything else. You carry it by that and the base.
You are looking at the incredible world of the microscope.
Quite like that?
We're all familiar with a magnifying glass.
He looks hungry, huh?
It enables us to see things much larger than they really are.
Sweetie?
Have you ever wondered how it is that magnification occurs? These students are about to find out by placing a book behind a glass globe filled with water. The book's text appears larger because of the curved surfaces of the globe. Ancient peoples used globes filled with water as the first magnifiers. They may have also used crystal balls as well. Much more practical as a magnifier is a thick piece of glass with curved edges called a lens. It is meant for examining just about anything in which you have an interest. Magnifiers come in a variety of styles. Some have a handle. These are very easy to use, but they usually have less magnification. This one has a magnification of 3. Its power is 3. It is meant to be placed a comfortable distance from your eye and adjusted until the specimen comes into focus. Other magnifiers are meant to be placed very close to your eye. These usually have a smaller diameter and a greater magnification. Some magnifiers are meant to be placed directly on top of an object. Others are part of containers. And still others offer a choice of lenses and magnifications. Regardless of the type of magnifier you use, observation should be made in bright light for better visibility, using an illuminator when necessary for dark objects. Here's a trick. Place a second magnifier above the first, and it will increase the overall magnification. In this example, the first lens magnifies the object 3 times. The second lens magnifies that image another 3 times. The overall magnification becomes 9. That is essentially how a microscope works, combining lenses to increase magnification far beyond that of a single lens. A microscope you might already be familiar with is a stereo microscope. Objects are placed on a platform called a stage. Solid objects are illuminated by a light from above, while thin or transparent objects can be illuminated from below. The lenses on a microscope are referred to as the eyepiece lens, there are 2 on a stereo microscope, and an objective lens, which often has changeable magnifications. The microscope is focused by turning the focus knob. Stereo microscopes usually have a maximum magnification of around 40. At this power, you will see that a smooth sheet of paper isn't so smooth at all. And neither is the surface of an egg. That a common mineral can resemble the surface of Mars. That a fungus can resemble a bird's nest. And mold a forest. You'll marvel at the pattern of ridges in your fingertips and discover that a picture is nothing but a sequence of dots. You'll find the hidden statue of Lincoln on the back of a penny and explore the tiniest detail in a minuscule fossil. The veins of a tiny leaf, pollen grains in a flower, the sharp points at the end of a wheat plant, nature's symmetry in a beautiful shell, the tiny scales in a butterfly's wing, the camouflaged eggs of an insect, the bizarre face of a walking stick, its strange mouth, and you'll marvel at a dish full of tiny brine shrimp. As powerful as the stereo microscope is, much more powerful is a standard light microscope. Most student light microscopes usually have a single eyepiece lens. The objective lens is usually located on a rotating turret with several other lenses. Multiple objectives provide a variety of magnifications. This objective will magnify an object 4 times. Its power is 4. This objective will magnify an object 10 times. Its power is 10. Finally, this objective will magnify an object 40 times. Its power is 40. The eyepiece of a typical student microscope usually has a magnification of 10. When you combine a 10-power eyepiece with a 4-power objective, the overall power or magnification becomes 40. This is a brine shrimp magnified 40 times. At this power, you can get an overall view of an object. When you combine a 10-power eyepiece with a 10-power objective, the overall magnification becomes 100. This is the brine shrimp magnified 100 times. Now we can see details we weren't able to see before. These are its eggs. When you combine a 10-power eyepiece with a 40-power objective, the overall magnification becomes 400. These are the brine shrimp's eggs magnified 400 times. Each magnification teaches us something new and different about a specimen. The microscopic world is waiting for you to explore. Your class may have a selection of specimen slides. These are things that have been professionally prepared for study. Specimens are very thin so that light will pass through them, and some are stained or treated in chemicals so that you can see them more easily. Glass slides should be handled carefully by the edges. Select one you wish to examine and place it on the microscope stage beneath the stage clips. The specimen is illuminated from below by either an illuminator or a mirror. The mirror should be angled toward a source of bright light, such as a lamp or a window, or as in this case, an illuminator. Mirrors should never be angled directly at the sun. Many illuminators have a knob that controls the brightness. Your eye should be placed directly above the eyepiece and a little away from it. You should keep both eyes open so they don't get tired. The large focus knob is used to bring a specimen into rough focus. The small focus knob is used to bring a specimen into fine focus. You may also have to recenter the specimen, which can be tricky because the specimen appears to move in the opposite direction you move it. Recentering is easy if your microscope has a mechanical stage. The microscope is a delicate instrument and should be handled with great care. You should never touch the glass in the lenses. Finger marks can damage a lens. Lenses should only be cleaned if they're dirty, using lens cleaner and lens tissue, rubbing with a gentle circular motion. A microscope should only be carried by its arm and base, and when not being used, a microscope should be replaced in its storage case or protected with a dust cover. Most student microscopes are set up for bright field illumination. This is the head of a crane fly shown in bright field. The background is bright. That's why they call it bright field. Your microscope might also have dark field illumination. Here the specimen is set against a dark background. Both methods of illumination have useful applications. The best thing about microscopes is what they can do. They open an entirely new world unseen by the eye alone. This is an ordinary ant magnified 40 times. This is the larva of an antlion. It uses its fierce-looking jaws to snap off the heads of ants. This is the larva of a moth. This is a fruit fly, commonly known as a gnat. This is the head of a common housefly. The multifaceted eye of a crane fly. The mouth parts of a grasshopper. The clubbed antenna of a beetle. Compare it to the feathery antenna of a moth. This is the rear leg of a honeybee, showing its pollen basket. And this, the elegant wing of a cicada. This is a chicken embryo at 33 hours. The fetus of a cat. The head of a newborn mouse. And this is its heart. This is a cross-section of a pine stem. And this, a tiny water organism called a daphnia. These are salt crystals magnified 100 times. Many household chemicals are crystals, and after they're dissolved, some form beautiful patterns as they reform. These students are about to discover that some of the best specimens can be found at a local pond. Back in the classroom, an eyedropper is used to deposit a drop of water onto a glass slide.
What do you cover it with? A cover slip.
The slide is then covered with a cover slip. It is then placed on a microscope stage beneath two clips. Mecium. They're feeding on pond debris. In dark field, you can see their bodies take on the color of the green algae they are eating. Paramecia move by beating their cilia, which are hair-like strands visible here along their outer edges. This one is called stentor. If you look closely, you can see the cilia that line the outside of its large mouth. The cilia are used in swimming and food gathering. This Vorticella uses its cilia to create water currents that pull in food. Larger organisms like these green algae get caught in the current as well, but are not consumed. This Dileptus feeds by waving its trunk back and forth through the water, hoping to draw in organisms. The Daphnia is a common multicellular organism you find in pond water. It moves by flicking its large antennae. The Planaria is a common flatworm you might run across. The Hydra has tentacles lined with sharp barbs that attach themselves to prey. Once caught, the victim is then stuffed into the Hydra's mouth, located at the base of its tentacles. The organisms you see in a drop of pond water may look strange, but they share much in common with you and me. Here's how you can prove it. Scrape the inside of your cheek with a toothpick. Dab this on a glass slide and cover it with a cover slip. Then place this under your microscope. As you can see, the inside of your mouth is made up of cells. A cell is the basic unit of which all living things are composed. Your entire body is made up of cells. Billions of cells that are not that much different from the ones in a drop of water, and the microscope proves it. Now try this. Scrape a little material from your teeth using a clean toothpick. Dab this onto a glass slide. Add a cover slip, and place this under your microscope, set to the highest power. You should be able to observe plenty of bacteria. They're the tiny specks jittering about between the cells. Bacteria are among the world's smallest organisms. These bacteria are relatively harmless. Some, however, are deadly. During the Middle Ages, millions of people died from bacteria-caused diseases. Because they could not see these organisms, people had no way of knowing bacteria even existed, let alone were the cause of disease. Even when the first microscopes were developed in the 1500s and 1600s, they were not very powerful and were not capable of seeing anything as small as bacteria. Then later in the 1600s, a Dutch microscope maker by the name of Antony van Leeuwenhoek developed a powerful type of microscope which he used to observe a variety of everyday specimens. These included insects, protozoa, and these, bacteria. He called them little animals, but he didn't know what they were. It wasn't until the late 1800s that the famous French scientist Louis Pasteur first proved a connection between some forms of bacteria and disease. Since then, cures to hundreds of diseases have been found. Today, the microscope continues to be the principal tool used in medical research. When cures to today's diseases are found, it is certain that the microscope will have played an important role. Both the traditional light microscope and the electron microscope, an instrument that uses electrons to magnify rather than light. Here, a tiny fruit fly is being prepared for study. It is placed on a small mount with a sticky surface. This is placed within a glass chamber, which is then covered with an airtight lid. Air is removed from the chamber to create a vacuum. Electricity is used to coat the fly with a thin layer of gold. The chamber glows purple during the coating process. The fly is then removed and taken to another room. Here, the microscope chamber is opened and the fly is placed inside on the microscope stage. The chamber is closed and a pump is started to remove all air from the microscope, creating a vacuum condition. Dr. Steve Barlow explains the difference in microscope types.
A lot of people wonder what's the difference between a light microscope and an electron microscope. Light microscope, as the name suggests, uses light to illuminate your sample, whereas the electron microscope uses electrons. A light microscope can magnify about 1,000 times normal size. The scanning electron microscope can magnify about 100,000 times normal size, or about 100 times more than a light microscope. This more powerful microscope allows us to see more detail in our samples.
The microscope stage is moved by control knobs on the outside of the chamber. Another knob controls the magnification, and another the focus. The image is displayed on a monitor, and while the picture is only in one color, it is nothing short of awesome in its detail. Here we're zooming in on a sensing hair on the fly's head. Now we're zooming in on the fly's eye, and we can see it is made up of thousands of smaller eyes and dotted with sensing hairs. And finally, we zoom in on one of the fly's legs and discover claw-like feet that enable it to cling tightly to just about any surface. These are viruses. They're responsible for some of today's deadliest diseases. Smaller than bacteria, they cannot be seen with a light microscope. And that's one of the reasons why electron microscopes are so important, to help us learn more about viruses and the diseases they cause. In this program, we have seen how a curved piece of thick glass called a magnifier can be used to magnify a variety of objects, and enables us to observe the world in a way we couldn't otherwise do. When one lens is combined with another, it increases the overall magnification. That's why stereo microscopes are more powerful than magnifiers and enable us to see objects in even greater detail. With stereo microscopes, objects can be illuminated from above or below. Standard light microscopes are even more powerful than stereo microscopes and enable us to see incredible detail in even minute objects. Standard student microscopes usually have a single eyepiece lens and a rotating turret that holds at least 3 objective lenses. Specimens for a light microscope have to be very thin so that light will pass through them. Specimens are illuminated from below with an illuminator or a mirror. The microscope is focused using either the coarse focusing knob or the fine focusing knob. Most student microscopes are set up for bright field illumination, but some may also have dark field illumination. Both methods have useful applications. The microscope has contributed to the cures of countless diseases and is today an important research tool, both the light microscope as well as the electron microscope. The microscope is also a passport, not to exotic foreign countries, but to an incredible miniature world.